Okay, well, it's my absolute pleasure to introduce Dr. Sanjay Srivatsan, who is, currently assistant professor over at the Hutch. Um, he did his undergrad at UC Berkeley and then did his PhD, his MD PhD at the University of Washington. That's where I met Sanjay a very long time ago. Um, he was one seat over in the lab for a very long time. So I think the way that I can, he went on to do some other stuff, like a very successful postdoc with Jason Urey and David Baker, and went on to start his own thing now at the Hutch. I think when I want to do a little bit more of an introduction to say that I think of all the scientists that I've met throughout the years, I think Sanjay is by far one of these people who just exudes creativity and kind of novelty in all of his work. And you know, sitting just sitting next to him was one of the best experiences of my science career. Just sitting there and drawing out on windows to see like, what can we do? And if we can't get it to work, how do we get it to work? And are you sure it doesn't work? I think it can't work. I think those are the types of conversations that are just incredibly fun to have. And just following his work after finishing his PhD has also been incredibly exciting to see all the new directions. So I'll stop there because I could go on for a while and it won't. But it's my pleasure to introduce Sanjay, and I know it's going to be a great talk. So, up to you.
Um, Thank you all for coming. And thanks, Jose. Like what he's describing there about sitting next to each other, that goes both ways. I think of Jose as one of my main scientific mentors. And today I'm going to be telling you about stuff that we've been working on for the last 2 years, focused on this new capsule technology and focused on whole genome single cell sequencing. So for a second, I'm going to zoom out and tell you a little bit about what our lab really finds as the central driving motivator for what we do. And it's this amazing fact that genomes encode self-assembling systems across a very disparate set of length and time scales, right? So here you have a mycoplasma here on your right versus a picture of the human organ system on the left. And both of those are encoded by sequences that self-assemble and form either in the course of minutes or in the course of years. And this is just a remarkable fact. And people have been trying to get at these kinds of, like what makes up an organism by doing single cell sequencing. And specifically, what we've seen over the last 10 years are all of these atlases that have formed, where you take an organism, you break it up into its component parts, and you ask, what are all those component parts actually doing? And by tracing this, set of lineages over time, you can see how the organism unfolds to some extent. But in reality, the kinds of snapshots that we'd really like to see are more like this one. So this is a tree core. And what's really remarkable about this is that just in this one picture, the life history of this organism is basically seen in many different aspects of looking at those tree rings. So you can tell the age of the tree by the number of rings. That's the thing people know. You can even tell things like the growth conditions, like how wet of a year was that? Or was there wind on one side? Do you see like differential spacing between those rings on one side of the earth versus the other? You can also see whether there's fire damage during that year. And in some ways, this tree core represents a complete molecular, or not molecular, a complete organismal history of this tree over time. And we would love these kinds of things, to know these kinds of things about cells in our human body. But what happens in our bodies is very complex. And to drive this point home, I always show this video from Kate McDowell, which was shot in 2018. So this is a gastrulating mouse embryo. And each time you see a single dot kind of light up there, that's a single cell division that's happening. And every one of these cells is being traced over time to actually form this gastrulating embryo. One thing to note is that capturing this video was, at the time, a technical feat. You had to build a special microscope body. You had to build adaptive optics that never existed before, and build software that had never existed before either. So this was a tour de force in kind of creating this moving recorded lineage of of this organism, but there's fundamental limits to how far this can go. So after this point, the organism becomes opaque, and you start to lose the ability to actually like retrieve light that you're putting into a system. So imaging can only take you so far. And really, if you want to go further, what you would really love to do is look at a cell's genome. So in some sense, the genome also contains a record of historic events in a cell. So it's estimated that there's 2 to 3 somatic mutations that occur during in every genome per cell division. So within all of us, every single cell effectively has a different genome. And then it's estimated that there's about 100 to 200 new mutations per diploid genome per generation. So that means when you go from mom and dad to child, you're passing along about 100 to 200 new de novo mutations. And because this is happening at such a regular rate, in principle, if you could grab every single one of these mutations, you should be able to reconstruct this whole tree. Secondarily, not only does the genome contain these errors, it also contains a set of mutations that are contingent upon what happened in the organism's history. So these are mutation spectra. So these are these trinucleotide mutations that occur in different kinds of contexts. And folks have worked to deconvolute mutations into these spectra that correspond to different things that happen during an organism's lifetime. For example, this spectra here called SBS5 is just a natural thing that happens over time as CPGs deaminate or demethylate. C to Ts are another example where you have UV radiation that will cross-link two thymidines, forming a thymidine dimer, and the repair mechanisms in our cells forms this very specific C to T mutation that can be recovered in the genome. So in some sense, if you can read the genome out perfectly, you actually already have that history of events that occurred over time and the lineage of those cells as they divided. And so a long-term vision, I think, for this field is basically, if you can faithfully sequence the genome, that would facilitate both lineage reconstruction, as well as the decoration of that lineage with historical events. But right now, we're really lacking tools in 2 fronts. One, on the library prep, how do you make thousands, if not millions, of single-cell genome libraries? And second, our sequencing accuracy needs to get better to the point where we're seeing mutations more often than sequencing error. And I'm going to talk to you about this first part, but there's really strong work happening in the private sector to make me think that the second part is just down the pipe. And so with that, I'm going to tell you about 4 different things today. I'm going to tell you about this capsule technology that we've been working with that really powers this. I'm going to tell you about how we can grow cells in those capsules, how we can perform single-cell genomics, and some lineage tracing we've been doing in this vein of counting somatic mutations. One thing to note is all of this is in close collaboration with Atrandi Biosciences, which is a small startup company in Lithuania. So all the reagents I'm going to tell you about here are commercially available, and you should be able to get set up with it right away. And then these are the 4 folks in my lab that led the study, and this works all on bioRxiv. Although I will be showing you stuff today that hasn't been in the preprint since it's been updated. All right, so I always like to start off with this really basic thing. How do we make measurements from single cells? One of the first ways that people did this was they put single cells into different wells, and then they would mark those cells with different nucleic acids. So for example, if you mark all the RNA molecules with this green barcode versus this pink one, you know that all the green barcoded molecules came from the green cell versus the pink cell. And this is 2012, 2014 era. When I first joined the Trapno lab, Cole had just done a fluid IMC1, and we were so excited about the ability to do 96 single cell transcriptomes at the time. That was just amazing. The next big shifts came in 2015, where Alon Klein, the Dropseq folks, and then the commercial effort at 10x basically broke this down into the problem of separating reactions into emulsions. So on the left, you're going to see single cells like this one kind of flow in and unite with gel beads that mark each of these containers as separate separate containers effectively. And what that allows you to do is kind of compartmentalize within each of these emulsions a single cell and its nucleic acid. One of the downsides of this kind of chemistry is that, unfortunately, although you've compartmentalized things, all your molecular biology has to happen in that one picoliter volume. So it becomes really hard to optimize new reactions because you can't add new reagents and you can't take away certain things. So you can imagine like one set of conditions that's good for one kind of reaction may not be best for the next reaction. And unfortunately, you have to break apart this compartmentalization that you've done to kind of introduce that next step. And this is where I make this distinction between emulsions and capsules. So emulsions are on top. These are phase-separated liquids comprised of generally water and oil. They can be split, merged, and sorted, but that is actually quite hard and requires specialized machinery. And the main thing that I want to highlight here is that you can't introduce new material without actually breaking that compartmentalization. Capsules, on the other hand, are comprised of a thin hydrogel surface. They can be split or merged, but they can be sorted, and you can introduce new material without breaking them. And I wanted to mention that this isn't just Trondi's effort. There's actually 3 different groups, including the Klein lab and the Mazudis lab in Lithuania, that have preprints out on this capsule technology. And each of these groups do different kinds of things. We focus on whole genome, but if you want to look at like transcriptomes or other kinds of things, I'd refer you to these 2 papers. But one thing that I am confident in is that this is basically the next step in the evolution of single cell genomics. So just like with emulsions, what we do is we make emulsions of a core and shell polymer with microfluidics. So here what I'm showing you is a diagram of the microfluidic chip, where you have fluorous oil that's pinching off a shell and a core polymer that are mixing in each of these emulsions. So next, once these emulsions form, the components phase separate. So you have like a fluorosurfactant on the outside. This yellow here is oil. And we expose it to blue light to actually initiate a photoradical process that actually polymerizes the hydrogel. Then you can strip away the emulsion or the surfactant and actually put your gel into an aqueous buffer. And what I'm trying to diagram here is that small particles can come out while large particles stay in. I'll tell you more about the size of those particles in a second. But what's great is that because we use microfluidics to generate these capsules, we can make them at different sizes. So here, just by changing the device, we can go between 25 microns in size to 200 microns in size. And because of the composition of the capsule shell, which is a saccharide-based acrylate, you can actually add enzymes that will cleave that moiety and release all the components within your capsule. And so over the course of this video, enzyme was added and those capsules slowly dissolved. And the takeaway, if there is one from this part, is really the capsule is this hollow sphere with a porous wall. And you can really see that in this set of micrographs, going from the most zoomed out on the left. One thing I should also add is if anyone here has questions, this is the bonus of coming in person, just stop me and ask, raise your hand. I'd rather answer it while the slide is up or while we're talking about that rather than having come back to it in the future. So you're probably wondering, okay, how porous are these things? The way we tested this is you put something inside, you wash the capsules a bunch, and then you see after every wash how much stuff is left inside. And what that looks like is effectively a gel on the capsule components. And so this is what the raw data looks like. And we can quantify this by fitting a exponential loss curve and plotting those beta coefficients. And when we encapsulate a DNA ladder, for example, what we see is that molecules that are under 300 base pairs slowly diffuse out of the capsules with every wash. And then we can do the same kind of test for proteins. And we can see that proteins that are under 300 kD can kind of readily equilibrate between the capsule and the outside. So when we first produced this data, one thing that really confused me was why are the mononucleosome and ribosome, which are such large multi-component protein complexes, why are they basically like coming in and out in this way? They should just be down here at the bottom. And one thought is, okay, these are actually complexes that are in equilibrium. They may be falling apart and coming back together. So what we did was we designed a set of what I call like molecular calipers. So these are de novo designed proteins that span different symmetry groups, and they all have different sizes effectively. So from D3 all the way to a icosahedron, which is 6-temer. And one thing to note is that these are super stable, like protein rocks effectively. And when we put those into our capsules and do the same test, in reality, this probably just reflects, the ribosome falling apart to some extent in our buffers, and they coming back together. But when you have really stable, large protein complexes, they really stay in. Things can also be washed in. So here I'm showing you a colony of T cells inside of capsules, and we put anti-CD8 FITC in the solution. And as time goes on, anti-CD8 kind of goes in and it actually stains those. So the takeaway from this part really is that SPCs, these capsules, can retain large biomolecules, and they really allow for the diffusion of small molecules into the capsule.
You can change the thickness of the shell?
Yeah, so the question was, can you change the thickness of the shell? And yes, to some extent, by altering the flow rates. But what we've seen empirically is that that doesn't really change the diffusivity and other properties of it. So we actually think that there's like, it's probably more complicated than just pure thickness.
The kinetics should change, though.
The kinetics should change, yeah. Yeah, but at equilibrium, it's more or less.
There's no contribution of charge as well in terms of.
That's a question I always get, and I think that's something we need to like nail down. We haven't done that yet.
Nucleosome and ribosome.
That might, yeah. Yeah, but for the ribosome measurement, what we did was like for every protein component, so like if you run a ribosome out on a gel, it just makes a ton of bands. We calculated for every band.
Oh.
And that's where I'm thinking like, okay, like maybe some of these are just. But those are pretty charged as well. Yeah, so I'm not, we need to really like look into that more. Okay, so these properties I told you about, the ability to let small things in, also allows you to grow cells in capsules. So here I'm showing the same microfluidics video, but here you see the little cells zipping in and going into those emollients. And so what you can do is you can put like a single E. coli marked by that red arrow there, and you can watch it grow over time to form this completely filled capsule. And we know that these are clonal because what we can do is encapsulate a mixture of red and green bacteria into the capsules and ask whether they follow the Poisson distribution, which we expect. And what we see is we see mostly red or mostly green capsules with the occasional ones that are mixed. So we're pretty confident these are clonal. And this doesn't only apply to bacterial cells, it also applies to mammalian cells. So here's a micrograph showing Hutch-3 T cells after 8 days of growth. And what you can notice is that like a lot of these cells start to fill and even start to stretch the boundaries of the capsule. And up here, you'll even see one that's kind of broken out. So that is one feature, like as time goes on, these cells will eventually kind of break through. This doesn't only apply to 293 T cells. We've basically tried it in many, all these are suspension cell lines, in many suspension cell lines and primary cells, including HSCs and primary T cells. And what we see is that we can actually find conditions that allow you to grow all of these different cell types. And the main challenge here was basically quenching radicals that are formed during that radical generation process so they don't harm the cells and allow them to actually survive and go on. I'm going to show you some more data about how we can use some of the cell culture to do single cell genomics better. So this permeability effectively facilitates the growth of small clonal batches of cells at a scale that's quite large. So just to put that in perspective, this is like effectively 250,000 clonal cultures happening in a single 6-well plate.
Destroying cells, the capsules affecting glycolysis is the transcriptome affected.
Coming in.
Is destroying the cells, the capsules, does that affect them in any way? The transcriptome?
Yeah, so that's like one of the reviewer comments, like what happens in that case. And when we do like RNA-seq of T cells, what we see is that the T cells grow in capsules have a lot more like cell adhesion or cell like we are next to each other kinds of transcripts and Go terms relative to like cells that's grown in culture. So there's clearly some effect of just growing cells near each other, but at a high level, they look like T cells. I think there's like 500 EEGs. Take that for what it is. Okay, so single cell genomics. So I mean, like this whole first part, I've just told you about how you can put small stuff in solution. You can have a big thing in your capsule, and then you can do reactions. And that's effectively what like all whole genome amplification and nucleic acid amplification is, right? It's small primers, small proteins that can diffuse in and kind of do your things. So we tested this by doing PCR in capsules. So here I'm going to show you a set of micrographs where the top row is the bright field, the bottom is DNA. These are then cyber stained with cyber gold. And what you're going to see here is on this bottom panel with different amplicon lengths, we can get PCR of, in this case, a MyoD1 locus in all these different capsules. So this is going off of a single genomic copy at a fairly high efficiency. We can also run these out in the gel and ask like, how specific is this product? And so here I'm showing you 2 lanes, and I'm showing it to you in this way where if you're looking at the square with the hollow circle, we're looking at the supernatant. And when we're looking at just the filled-in circle, that's within the capsule itself. So there's really 3 things I want to point out to you. 1, the genome's at high molecular weight and it's only found in the capsules. 2, there's only primer dimers that are found in solution. And that's because the primer concentration is really highest in solution there. And then 3rd, and most importantly, I don't have it written here, but you can see the product in the supername as well as in the capsules. And we thought initially going into that would be like length restricted, but actually it turns out that almost at every single molecular weight, you can see product in both the capsules and in the supername. And I have theories for why this is, and I can tell you about them later. But the first thing you'd probably want to know is, okay, like if this happens, does it go into empty capsules and contaminate things in a way that we would care about? So you can do the inverse experiment where you have empty capsules and then you have templates and primers in solution, and you can perform PCR a bunch, and then ask how many times you actually see capsules that have stuff in them. And at the low range, we can see some, like below 400. But as this goes up, we see very little. And we follow this up with both, nested PCR and sequencing, and this does seem to hold out. So briefly, okay, so in the beginning, I told you about 2 different ways of making single cell measurements. There's well-based, emulsion-based, and then there's this 3rd way called combinatorial indexing, which many of you are familiar with, partly because of Jose, partly because it really is becoming a more mainstream way of doing things. And just to introduce this concept to you briefly, the idea is that if you take a container full of nucleic acids through a combination of barcoding steps, every nucleic acid in that container will share the same set of combinatorial molecules. And so when you sequence the combination of teal and red barcodes, you can say that, oh, all of those came from the same container. And really, these capsules are the best container ever for combinatorial indexing. And it's because they allow fairly arbitrary chemical reactions that can be performed on nucleic acids that we couldn't do previously when we were using the cell as the container itself. So for example, we can perform multiplex PCR in mixtures of cells. I'm going to breeze through this part pretty quickly. We can detect those 4 amplicons in 97% of cells, very sensitive. So for a single allele, where that's a heterozygous in a cell line, we can detect both alleles in about 85% of cells. And so this is to say that, like, for, you know, for doing things like qPCR or multiplex PCR, this is a really effective way of looking at a bunch of sites across a large population of cells with a highly specific reason. Second, we can also amplify the whole genome, and that's really what we've been interested in, because, like I told you, we want to discover these somatic mutations, and we want to map them back onto the genome and ask where things are different. So we can do this. So this is the same set of micrographs that I was showing before: pre-amplification, post-amplification, same exposure, same normalization. These things just have tons of DNA in them. And one classic experiment that single-cell folks always do when they're first starting these things is they take 2 different species, often human and mouse. They mix the cells together. They perform their library preparation and sequencing, and then they map cells back to either a joint combination of the mouse and human genome. And the idea here is really simple. So if a read comes from the mouse genome, it should map to the mouse. All the reads from that cell should map to the mouse genome. And if a cell is a human cell, all the reads from that should map to the human cell. So this basically on-axis plot is exactly what you'd hope to see if you're seeing minimal leakage between your genomes, which is effectively what we're seeing. So the thing that we've really been interested in this area is, if you want to do single-cell whole genome sequencing, you really want to see every single site in every single cell. That's because, like, when you compare between cells, you want to be seeing the same parts over and over and over again. So you can really build a dense comparison between the same set of sites. So it turns out that sequencing a genome evenly is one of the hardest things to do, especially when it comes down to having a single template. So to assess this, folks in the field use this thing called a Lorenz curve, which I'm showing here. It's basically asking, for every read that I add, is that read unique? And in the ideal sample, you'll see a line that corresponds to this dashed line here. Meaning, so like of the cumulative reads I have there on the y-axis, what % of the cumulative fraction of the genome in my sample am I covering? So if every single read I'm adding covers a new part of the genome I've never seen before, then that's a perfectly, like, complex sample. And that's our ideal bulk sample. When you do actual sequencing, so if you actually sequence the bulk sample, you would get this line. So this is data that we reprocessed. So this field of single-cell whole genome sequencing has been progressing since 2006, where the first studies were done with this reaction called MDA. And what you can see here on the Lorenz curve is that as you add these final set of reads, you're not really covering that much more of the genome. So that means that these libraries are not really as complex as you'd want them to be, and they're biased towards certain parts of the genome. So that was in 2006. The Malbach paper came out in 2012. That was just a new way to do single-cell whole genome sequencing. In reality, it didn't seem to increase the complexity of these libraries that much. Then came a set of papers, including Leonti and PTA 2017, 2021. And what we're seeing is that those curves are effectively starting to get towards the bulk. So these are single-cell genomes that are being sequenced deeply, and we're starting to see the basically ideal distribution that you would see in an ideal gold sample. And then in light blue here, I'm showing you basically our version of doing primary template amplification, PTA, inside of capsules. And we're looking quite good relative to PTA and Neon 2. One way that people in this field also think about amplifying genomes is just letting cell divisions do the work for you. So if you can just allow a cell that only produces 2 to 3 mutations per cell division, do the replication, you're going to have a really low mutation rate. It's really even, and you can basically get out of it exactly what you wanted your enzymatic procedures to do. So we took our hematopoietic stem cells from cord blood and performed cell growth inside of the capsules. So going from day 0 all the way to day 8. And on day 8, we harvested these capsules, sheared them within the capsule itself, put sequencing adapters, performed common control indexing, and then sequenced the genomes. And when we do that, we can see that, again, the cell division in capsules is starting to basically get towards this ideal line. And so this was data that we sequenced like last week, and we just ran a couple NovaSeqs on this to really like give it the depth that it deserves. But I'm really excited about this because for 2 reasons. One, you can just like look at, so this is the raw coverage for 1 kb bins across the human genome. It's normalized per bin effectively. And so this is the primary template amplification of a hematopoietic stem cell. Just one note, this is actually quite good for single cell genome sequencing. You're seeing most bins have something in them. And then you're seeing small peaks, mostly at the telomeres where there's mapability problems. But when you look at something like the cell division kind of distribution, you're seeing an incredibly even coverage across the genome. And this really says that, you know, if you're allowing the cell to do the work, and if you have a cell line or a cell system that allows you to replicate and grow within the capsules, you can produce remarkably even library coverage across a single cell genome. And you can tell here that like the coverage on X is half because, you know, this is like a male, the cord blood came from a male person. So, okay, so building it up further, so these SPCs facilitate simultaneous, uniform, sensitive genome amplification, allowing you to barcode basically thousands of genomes and sequence them at once. Cool. Okay, so now I'm going to tell you about how we can apply those ideas to do some lineage tracing. So I showed you this picture here before of our tree core. You know, this is like the ideal that we're trying to get towards. And this is like really quite a diverse field called molecular recording. And people in the molecular recording field, I mean, this dates back quite far. They think about basically like, how can I record these events like lineage, like stimuli that are coming from the cell, by writing into DNA. And the main way these folks think about this, I'm just going to summarize it in a set of cartoons, is if I have a single locus shown by these 5 boxes, I can use some molecular process like CRISPR-Cas9 or some other kind of DNA writing technology to basically add random edits to that cassette in a time, like over time, so that as time goes on, each of these diversifies. So what I'm showing you here is the true set of sequences that happened where in this cell, the green and blue mutations were added, and this cell, the red one was added, and then on to the next generation. And then the idea is you can reconstruct the underlying tree just based on this one locus. But in our view, there's some real fundamental problems with this. And the main one that we've been really thinking about is that this process of writing into this locus is not really pegged to any real sense of time in the world. Meaning, you'd want two things. One, you'd want the writing process to be continuous so that it happened with every single cell division. And then two, you'd want to have some marker for the amount of actual wall time that has passed. So like, let's say that it's been 2 days. You would love to look at these loci and infer that it's been exactly 2 days since you started this experiment. And like I mentioned before, the genome actually already has this information within it. What these approaches do is that they let you look at one locus with very efficient sequencing and infer a process that's happening genome-wide. But our bet is that as genome sequencing becomes cheaper, as these methods become better, instead of looking just at that one locus, you'll be able to look genome-wide and infer both these clock-like mutations, as well as these mutations that are scattered through the genome. And okay, so it would be a fool's errand if I just tried to go off those 2 to 3 mutations per cell division, just because that's like looking for a needle in a haystack, effectively. So what we've been doing is using 2 different, or really 1 different mutant of polymerase called polymerase epsilon. And just for reference, polymerase epsilon, polymerase delta are 2 genes which basically like copy either the leading or lagging strand of the genome during cell division. And in some of the TCGA papers, it's been well documented that certain mutations of Pol E and Pol D are ultra-mutator alleles, meaning they increase the mutation rate of cell division by 1,000-fold.
The largest.
What's that? Proofreading? It's in the proofreading domain. But what's actually amazing is that these aren't proofreading null mutations. So you can have proofreading knockout mutations. These knockout proofreading and increase mutation rate beyond that. And there's a specific structural reason for why people think that is. But this is even higher than proofreading knockout. And another amazing quality of this mutator allele is that it makes a trinucleotide spectra that's very specific, right? So even in a genome that's awash with different kinds of mutations, you should be able to pick out that this TCT to TTC mutation is the one that you're actually. So what we did was we first installed this polymerase epsilon mutation called P286R into K562 cells. These 2 plots just show that the cells are quote unquote like healthy and normal. These are K562 cells, so there's really nothing normal about them. And that their allele frequency doesn't really seem to go down as a function of time. So they're being stably maintained in this population. When we just sample sequences from each of these and perform bulk sequencing, we can see that the mutations in the genome per million reads goes up monotonically, both at locations around the genome and at these specific sites. And so then we performed what we thought was a really simple bottlenecking experiment that turned out to be like much more complicated than we expected. So what we did was we took 1 to 2 cells and we bottlenecked them. And then we allowed them to grow for 11 days and then performed the first split. And then we made this thing called a caterpillar tree, which is this thing that's shown on the left, where one of the populations was continually split over and over again until you got basically this structure here. So this is the underlying ground truth structure on the left. And now the question is, okay, if we do single cell sequencing, to what extent can we recover that structure? So what we did is we generated about 1,600 single cell whole genomes from this cell line. We, just as a metric of success, we like recover nearly every SNP that's found in the K562 reference genome. When we call mutations, this number was probably going to go down as we sequence deeper. We can see about 22,000 de novo SBS on average. And I think I need to update this number here, the 74,000. And one thing to note is that most of these are singletons, meaning that they're only seen a single time in the whole, just let me finish this. Most are singletons, which means that they're only found a single time in the whole population. And 873 are high quality de novo variants, meaning they're found, the same SNP is found multiple times in multiple cells. And then one more thing to add, just to add to your intuition for this, in these systems where you're exponentially accruing errors with every cell division, half of all mutations occurred on the last cell division. So I'll let that sink in for a second, and then I'll take the question. So like you would expect half of all mutations to be single.
So there's a question on the chat from Eva Soler. Since I may have missed this, but are PCR to these mutations or error considered when analyzing the sequence, or how many PCR cycles are done? I guess how prevalent do you?
Yeah, so there's a few sources of just like technical error. So for example, like when we do whole genome sequencing, we add NaOH, right? And that can basically like introduce certain kinds of mutations. PTA or MDA, they use 529, that adds mutations. When we perform the final PCR library prep, we do like 15 cycles of PCR, so there's mutations there. So these are all accruing in different ways. We try to filter those out just by analyzing the singletons on their own. But often, as you sequence deeper and deeper, those will fall away. And there's certain kinds of signatures you can look for those as well.
Okay, I'll add one more question. So from Joe Bell, how does your whole genome sequencing coverage differ when you perform PTA on one cell versus the growing cells within the capsid? I guess how you miss coverage when you only have one cell per capsid?
Yes, that's coverage. Well, we have like the known reference genome. So like our NovaSeq, right? So like all the data I've shown you is a less than 1x. And at that sampling frequency, like it's very hard to assess these things very rigorously. So right now we're trying to get a 30x per cell. So we can actually like put them heads to head. So right now at this low depth, they look pretty similar. But if you ask me like that, I would say that the single cell division is going to be a better way of achieving evenness and like even genome coverage. Okay, so I'm almost done here. So one thing to note, this is comparable with the number of cells in the C. elegans genome, which I will get to in the future as well. So first, like when we look at like the high quality de novo SDS, we see our expected deconvolution, where you see like 17% of our mutations are in this clock-like signature. We can also see our poly signature corresponding to 4% of the sites. And then we can see other SDS that are corresponding to K562's natural way of reproducing. So when we aggregate all of our mutations per group and then try to reconstruct the actual underlying tree, we can get a almost perfect reconstruction of the actual underlying structure with very good bootstrap support all the way down to the lowest clades. And with a sense of like somatic divergence, meaning that there's a sense for which like this was the first thing split and then so on. And like ideally in the future where we're trying to get to, we want to be able to resolve how many cell divisions have occurred in that cell for a given amount of time.
Sanjay?
Yeah.
Just to clarify, that means that you're not doing any live imaging while.
That's right.
Right,
Yeah, there's no imaging.
just be cool for the purpose of proof of concept with the number of divisions.
Yeah, so I can just say this now, so like because I'm almost at the end. Our version of that is we've made a worm, C. elegans, that has the Pol1, which is its version of Pol E mutation. And there, since we have the stereotyped lineage, and we can use the transcriptome to basically line it up, we know how many cell divisions have occurred in every single one of those cells. So we should be able to count in those single cell genomes how good our basically like cell division versus mutation rate calculation actually is.
So with like, for example, 30x coverage from sequencing, like deeper sequencing, like without the poly mutation, like does it allow you to like reconstruct this biology with just naturally?
No. Yeah. And that's purely dependent right now on the error rate of sequencing. So like Q30, which is like the thing that you can get from Illumina, it's like, you know, their highest guarantee. That means one error every 10,000 bases. The mutation rate in this system, or like in our cells, is 1 for every 100 million. So you need to get to like Q90 or something effectively before you start to like start to have sequencing technology where you can actually believe the basis of it.
And this is only the luminescence.
What's that?
Luminescence.
This is only a luminescence. Yeah, and you can get over that with like circular consensus sequencing. You can resequence, you can do duplex sequencing. Those are ways to get to like Q60. But if you can get to Q90, I think like that's where you're going to be. So like underlying that data, there's a single cell lineage tree. The reason I said that this is a complicated experiment is because by the time you get to day 11, you have such a large population size that your splits aren't going to perfectly concord into any set of lineages. So this is also why we're doing the worm experiment, where there's a really well-defined ground truth. But I will say, like, okay, we were like, okay, what can we learn from even this representation of the data? So we can run things like AnuFinder, which will ask like, what's the ploidy of each of these genomes? And what we see is like in K562s that have this polymerase epsilon mutation, we see an increased incidence for genome instability, meaning there's a lot more like duplication or triplication or chromosome losses and gains. And because we have the tree, we can now decorate it asking, okay, is this duplication happening in one cell and then spreading, or is it happening across the whole tree? And indeed, it seems to be happening, if you believe this underlying tree, it seems to be happening independently across the lineage over and over again. And this isn't a thing that we're just seeing here. Like if you look within the mouse that has this mutation, you can often find aberrant chromosomal abnormalities where cells either have whole genome doublings or whole genome triplings. And we really think that in some way, what all of this is telling us that cells contain some kind of intrinsic sensor that links genome duplication with mutation accumulation. And I think this would be the perfect place if anyone in this room or this audience like wants to do a CRISPR screen on this kind of thing. I really think that there's like a linkage between these two. Just because if you look in every single one of these publications about polymerase epsilon mutations, you see this link coming up over and over again. And so these are the things that we're doing in progress. So we have a mouse that has this polymerase epsilon. Surprisingly, these mice are like viable up to like many generations. Like one of our collaborators has them at the 14th generation, where they continue to of hets. So that what they do is they take a het and then they back cross it to like that Black 6 from Jackson Labs, and they've kept that going for over 14 crossings. So these genomes are really littered with just mutations from every single one of those generations.
So
Yeah.
you should look at the sense mutations, those generations where you have access to the
Yeah, that's like the kind of thing that would be amazing, because we think that it hasn't been generated yet, but yes, we will let you know when.
Yeah, I mean, that's the coolest part of it.
Yeah, because it's kind of like shotgun, what do you call it? I don't know, what do we call that? Yeah, just shotgun mutagenesis of the exome or whatever, the whole genome, with the constraint that you have to produce an organism next year. Yeah, so these guys have just like way higher mutation rates. They have way higher cancer rates. We're applying an RNA DNA protocol to trace tissues from both worms and mice in this context to really ask like how far we can push this. And then like the thing that always gets me really excited is like, can we really count cell divisions in vivo? And right now I think like that's my like North Star. So in summary, so these SBCs are really flexible containers for molecular and cellular biology. You can use them for really flexible and scalable single cell assays. So we've been developing molecular tools for amplifying single cell genomes at scale, but I really think that you could do anything that you can do in a test tube in these capsules. So, whatever you do, you could probably convert it to a capsule-based way. And then lineage tracing via continuously deposited mutations, I think is going to allow us for reconstruction of cell division resolved cell lineage trees. And this is like very much ongoing in our lab right now. And with that, I'll do some acknowledgements. My lab is about 8 people large, including myself. And we're always looking for people that are excited to do a postdoc or at any other stage of training. Dustin, Sam, David, and Ian led this work. And then Ropolis at Atrondi is one of my main collaborators. I would get in touch with him and Emilis if you're interested in this capsule stuff. Like I said, all of this is like completely commercially available, and you can do amazing things with it as well. And then my other collaborators and mentors in Seattle, and then our funding sources. And I'll take any questions.
Might be a little normal question, but as you accumulate mutation and then also start affecting the mutation rate, can that lead to complication in terms of what you're trying to do?
Yeah, so the question was basically like, if you're accumulating mutation rate, will that affect the rate of mutation and rate accumulation? And so when this, so in studying this polymerase, there's like a few different fields that do it. There's this one branch in yeast that looks for suppressors of mutation rate accumulation, meaning are there other genetic mechanisms to compensate for it? And often they find that one of the main suppressors is just revertant of that site back to its normal thing. So yes, in principle, things like that can happen. And in practice, I think that's actually a really cool area of research. I don't think we know yet. People do think that there are, in the yeast literature, they think that there are other mechanisms to suppress mutation rate that don't involve revertance, but no one's found what those are.
Sorry, I didn't think of this question. We're showing this graph when we basically had coverage of as against the bulk genomes. Did you stratify that plot by number of duplications of that genome for that cancer cell line? Like if it's a 6 cent, right, you're just going to get naturally better coverage of that cell. And I mean, your sequence, like you just have these are cancer cell lines, right?
Yeah.
You're going to have crazy amounts of this.
So,
Not saying bulk doesn't have that. I'm just kind of thinking how confident you are in the sensitivity. Yeah.
Yeah, so in this field, there's a reason that we use cord blood.
Okay.
And the reasons are twofold. One, this is the lowest mutation rate somatic tissue that's known to exist. For some reason, blood, especially the stem cells, don't get mutations. And then second, we've also just sequenced a bulk sample from this person just to make sure there's no ploidy differences. But in the cancer context, I definitely agree with you.
Yeah, sorry, I missed that.
Yeah, there's also this little marker over it as well. And I think, yeah, we want to, I think there was another question in the chat from before asking about like where the mutations come from. We want to benchmark against these low mutation rate tissues to actually know which mutations are coming from us and which mutations are latent in natural tissue itself. To just go again, Sonia. Yeah, it's good.
Let's do one. I'll come back to one.
Okay, one, so.
So absolutely stunning talk. Two quick questions from Ravindra. What kinds of cancer do these hypermutator mice get? And are there ways to change the semi-permeable capsules into impermeable ones? That's interesting.
Into what?
Impermeable. So, at some point shut it off again.
I'll answer the second question first. There's a really simple physical trick you can do of just vortexing the capsules in oil and re-emulsifying them. It's called particle templated emulsification. And that works really well. So you can basically like re-emulsify them really fast. Regarding the first question, it's actually super interesting. Depending on the life stage of the animal, you get different cancers. So like the first set of cancers that will kill these mice are thymic cancers. And it probably just corresponds to the amount of cell divisions that are occurring in the thymus and their reliance on polymerase epsilon to actually fix those errors. If the mice survive that, then they go on to get colonic and skin cancers. And then there's also this kind of, in medical genetics, it's called anticipation. So it's like what you see for Huntington's disease, where as generations go forward, you see the disease come earlier and earlier in time. So it turns out the F2s and the F3s and the F4s, as time goes on, it also depends on what the baseline mutation rate in the germline is. So if the mutation burden is so high in the germline, the first cancers you see are leukemias. And that's from this burst of fetal hematopoiesis in its infants. So there's a combination of like, what does it take to make an organism? Like, where are the bursts of cell division happening in which organs? And probably a mixture of what are the repair mechanisms and polymerases used in those lineages. There's some convolution of those two things that really determine it.
Oh, right. Yeah, I came back to it. So, this is mostly on fresh cells, right? Or broken cells. It's, you didn't do any, or try to maybe optimize any experiments with a trendy one, nuclei or frozen samples, or potentially or organoids and stuff like that. I go a bit more clinical route.
Yeah, I think like in principle, I don't see a problem with for the genome stuff, right? If you can make a single cell suspension, you can get it in here. You can do arbitrary chemical reactions. You can add denaturants like guanidine thiocyanate. You can add heat. You can add you can decarbonize things. You can add Pro-K to like really break things open. So like in my mind, probably with some optimizations that are always required for these things, this should work if you can get into single cell suspension. But yes, to answer your question, I'm only working on the easiest stuff right now.
Another question on the chat. So Greg, Tom, one last question. I think I recall seeing that you did this with E. coli, but are you able to grow two different cell types in one instance? Or something like a cell-to-cell interaction assay?
Yeah, so we've done that with like a T cell and a cancer cell that elicits it, and that is totally doable. I think the thing that the questioner has to think about is, and this is the thing that killed Dropseq, is that if you want to do like double loading, you run this thing called double Poisson, where if you just want one instance of both, you only get that in 1% of your population. But if you have responder cells or stimulator cells in every single capsule, and then you only have in 10% your T cells, for example, then you can kind of like skew these things to like that. So yeah, in principle, you should be able to do this.
Does the PTA imply mitochondrial genome? Does that mutate more frequently than?
Yeah,
Curious with that.
yeah, the mutation rate for the mitochondrial genome is like 1,000-fold higher. And like people have taken that approach to lineage tracing. The problem often ends up that, and you can see this if you like look at micrographs of dividing tissues. The distribution of mitochondria per cell division is very unequal, and it can be based purely on the kind of cell division that's happening. So for certain asymmetric cell divisions, like often like almost all the mitochondria are gone, or and sometimes 50-50, sometimes 40-60. And that just introduces a lot of like variation to lineage tree growth. But we do have the mitochondrial genome in here, and we are including it in our calls. But we haven't just used it alone to ask this question. Oh, wait, and then secondarily, maybe this is the question you're getting at. Mitochondrial genome uses a different polymerase to amplify it. So polymerase epsilon doesn't do the mitochondrial genome. I think it's like polymerase gamma or something.
And so to see, well, we're out of time.
Okay.
One more question.
Last question.
Yes, it was kind of similar to that one, because I was thinking, there are other like somewhat built-in lineage traces that people have used, which is mitochondrial DNA. Transposons, right, is the other one. And you can think as well in terms of colon cancer and microsatellite instability.
Yeah.
So I guess, have you thought about playing around with multiple inputs in terms of, how well can you capture microsatellites?
I think it's really hard. With short read sequencing, like the average microsatellite length is like long enough that you can't map across it with a single short read.
But in theory, you could do long reads.
Yeah. So like if you could do a complete genome assembly, that will give you so much more, because you can use indels, you can use like these like microsatellite instabilities. Yeah. We haven't. That's like it. I guess we've just been using SNPs.
Well, let's thank Dr. Srivatsan one more time.